Hello, my name is Jancy Ferrer. I'm an assistant professor and researcher at the Universidad Central del Caribe, and I will be presenting our most recent work showing that memantine and the 4R-Cembranoid share similar cholinergic pathways to induce neuroprotection. Memantine is an FDA-approved drug designed to treat patients with moderate to severe Alzheimer's disease. Studies show that memantine is an uncompetitive antagonist of moderate affinity to the NMDA receptor. However, memantine's therapeutic role cannot be exclusively attributed to its effect on the NMDA receptor. Several studies have demonstrated its effectiveness as a neuroprotective agent by its ability to inhibit nicotinic receptors. In this study, we show additional evidence that memantine's mechanism of action contains a cholinergic component. In our studies, the pharmacological profile of memantine's neuroprotective pathway was examined using ex vivo rat hippocampal slices. In this procedure, the hippocampal slices were extracted and left to recover in an artificial cerebrospinal fluid inside a 3-lane population spike recording chamber, shown to the left. After 1 hour of recovery, the initial population spikes were recorded. Then, the hippocampal slices were perfused for 1 hour with either memantine or inhibitors throughout the study. These inhibitors were added 15 minutes before memantine treatment. Afterwards, the treatment is washed out and all the slices are exposed to an excitotoxic 0.5 millimolar NMDA insult for 10 minutes. After another washout and recovery, a second population spike measurement is made. Finally, the percentage of population spike recovery after the insult is calculated and reported. In these studies, we will compare memantine to 4R. What is 4R? 4R is a small hydrophobic molecule that crosses the blood-brain barrier, and it has anti-apoptotic and anti-inflammatory activity. Patch clamp studies on neuronal cells overexpressing the alpha-7 nicotinic acetylcholine receptors have demonstrated that 4R negatively modulates this alpha-7 nicotinic receptor. In other studies, it has been shown that 4R also negatively modulates the alpha-4 beta-2 nicotinic receptor as well. Our group has shown that nicotinic modulation by the 4R-Cembranoid has neuroprotective effects in several models of neurodegeneration, including ischemic stroke, Parkinson's disease, and intoxication with organophosphates. Although 4R is not an NMDA receptor antagonist, we show in these studies that it has a similar neuroprotective mechanism of action to memantine. This statement was first supported by ex vivo hippocampal slices studies showing that memantine induced neuroprotection before or after an NMDA insult. But interestingly, in the presence of the NMDA receptor antagonist AP5, there was still a 30% of neuroprotection from memantine that was not dependent on the NMDA receptor. Other neuroprotective mechanisms we know exist are the cholinergic mechanisms. And in the 1990s, Parsons et al. Described how memantine could also have affinity to nicotinic receptors. These results were further supported by ex vivo hippocampal slices studies, which demonstrate that the neuroprotection of memantine against NMDA was completely inhibited in the presence of an alpha-4 beta-2 nicotinic receptor antagonist named DH-beta-E. These results have also been shown in previous studies for the 4R-Cembranoid. Finally, in the presence of the cholinergic vesicle release inhibitor called vesicle, memantine's neuroprotection was significantly reduced by 25%. Therefore, the cholinergic component of memantine was shown to be critical in its ability to elicit neuroprotection. This has also been shown previously in studies for the 4R-Cembranoid. To elucidate which cholinergic neuroprotective pathways memantine follows, we first tested the well-known mitogen-activated protein kinase pathway. To test this pathway, we added the MEK1/2 inhibitor named PD98059, 15 minutes before adding memantine. The neuroprotection by memantine was not affected when co-applied with the MEK1/2 inhibitor. This is also the case for 4R we have published. Next, we wanted to test if memantine's neuroprotection used the PI3K-AKT pro-survival pathway by blocking it with the PI3K inhibitor LY94002. Our results show that memantine mediated neuroprotection through this pathway as it significantly reduced its neuroprotection by 30%. Interestingly, we have published that 4R neuroprotection is blocked entirely when the PI3K pathway is inhibited. Therefore, both drugs use the PI3K-AKT pathway to mediate neuroprotection. In conclusion, having a similar mechanism of neuroprotection, but with a wider neuroprotective range, we propose that the 4R-Cembranoid is potentially a better alternative treatment than memantine to ameliorate the symptoms of Alzheimer's disease. Future experiments will be aimed to determine if 4R can provide neuroprotection in a tauopathy or an Alzheimer's disease mouse model. Finally, we would like to acknowledge our mentors and collaborators, Dr. Pedro Frischming and Dr. Vesna Eterovic from Neuroprotection for Life Company, Dr. Antonio Enrique Martins from UPR Medical Science Campus, Ms. Dinelli Perez, which is the electrophysiology technician at UCC, and Dr. Nadezhda Sabeva at UCC. Thank you for the research support granted by NIH and INDS NORD grant, and thank you all for your time and for watching.